Hello friends, welcome to Vidya Edu World, and our today's topic of discussion is genetically engineered insulin. Before studying how insulin can be made by genetic engineering, firstly we will see what are the roles of insulin in human body. As we know that insulin is a hormone which is made by pancreas. This insulin is essential for uptake of glucose from blood and conversion of it, that means conversion of glucose into energy, which is required for every cell of our body. So, ultimate function of insulin is energy production. So, it is utmost important for energy fulfillment of our body. Also, by this, it controls blood sugar level. Nowadays, diabetes is ever increasing disease in our community. This disease is characterized by elevated blood sugar or blood glucose level because pancreas does not produce sufficient insulin, or in some cases produced insulin is not used efficiently in our body. So both of these things, that is low production of insulin or its inefficient use, leads to the increased blood glucose concentration. So the scientific term used for this is hyperglycemia. You can see hyperglycemia means increased blood sugar level, and it is a typical diagnostic symptom of diabetes. So this hyperglycemia can damage vital body organs. Therefore, it must be treated to control blood sugar level at normal level. Then the question arises how to manage diabetes or how to control the blood sugar level. As we saw earlier, one reason for diabetes is inadequate insulin production. So to fulfill body's requirement for insulin, we can take insulin injections externally. At this point, next question arises, what would a diabetic patient do if enough human insulin was not available? Because number of patients are increasing day by day due to unhealthy lifestyle. So one solution can be use insulin isolated from other animals, as animals also produce insulin for glucose metabolism. Then again, you might have a question in your mind is that would the insulin isolated from the cattle or animals be just as effective as that secreted by the human body, or can it cause immune response in the human body? Because though the function of animal insulin is same in humans and animals, that means control of blood sugar level and energy production, but the animal insulin is foreign protein for our body which can cause immunogenic response exactly in the same way when new pathogen or antigen enters in our body and our body gives immunogenic response against that pathogen. Then the big question is that what to do? Now imagine if we insert human insulin producing gene into the bacterium and this genetically engineered bacterium could make human insulin. If this happens, then we can grow bacterium and can harvest insulin as a product from them to meet our requirement. Before the discovery of this genetically engineered insulin, the insulin used for diabetes was extracted from the pancreas of slaughtered cattle and pigs. And as we discussed, it may cause allergy in some patients as this insulin is from animal origin. That means it is foreign for our body. If we see the biochemical structure of insulin, basically it is composed of two short polypeptide chains that are chain A and chain B. And these two chains are linked together by disulfide bond. Also, the extra stretch of polypeptide chain C is present. In mammals, including humans, insulin is synthesized as prohormone or proinsulin. So prohormone is basically a non-functional hormone which needs to be processed before it becomes fully mature and functional hormone. So in this diagram from NCERT book, you can see proinsulin is composed of three chains, chain A, B and C. When this proinsulin is processed, then extra stretch of chain C is removed, whereas the chain A and chain B remain together by disulfide bonds. So this chain A and B together is called as functional insulin with the removal of chain C. It is quite difficult to process this proinsulin in vitro, that means in laboratory, to remove C peptide from them and obtain fully functional insulin by recombinant DNA technology. So the process is somewhat simplified when in 1983 an American company Eli Lilly discovered a new way of preparation of functional insulin by genetic engineering. So what they do, instead of transferring entire gene of insulin in E. coli bacteria through plasmids, they transfer only gene for synthesizing A peptide chain and B peptide chain separately in bacteria using plasmid as a vector. So, thus the chain A and B were produced separately, they are extracted and these two separate chains combine together by creating disulfide bonds to form human insulin or fully functional human insulin. So this is all about the outline of the genetically engineered insulin. If you like the content, then like, share and subscribe my channel. Have a good day.